Neurogenesis is the process of creating new neurons. And for decades, textbooks have said that there's no neurogenesis in the adult human brain. No new neurons. Your skin cells turn over every couple of weeks, with new cells coming in and old cells dying off. But your neurons, those are with you for life. Now, in some ways, this is a good thing. If we were constantly turning over new neurons, then how could we ever have stability in the way we process visual information, or the way we store memories, or the way we create our personality and our sense of self? Of course, there are big disadvantages to stability too. If we never create new neurons, that means it's going to be that much harder to recover from any kind of brain damage or any kind of neurodegenerative disorder. It might actually be a good thing if we could grow some new neurons sometimes. Now, it's also been known for a long time that humans are fairly unique in our lack of neurogenesis. Rodents, for example, are known to exhibit neurogenesis in the dentate gyrus granule cells of the hippocampus for their entire lifespan. And this is a particularly interesting place to exhibit neurogenesis, because this is one of the key areas that first shows dysfunction in the development of Alzheimer's disease. So if humans happen to have neurogenesis in this same location, then it might hold promise that we could somehow augment that neurogenesis in some way to help be curative for Alzheimer's disease. And there was hope for this kind of idea because in 1998, a team spread between California and Sweden published a paper where they said no to all of those decades of research. There actually is neurogenesis in the adult human brain. We just missed it. Now, this was huge news. It opened the door suddenly to the idea of regenerative medicine with new neurons as a key part of rebuilding the brains of injured individuals or people facing neurodegenerative disorders. But it's been very controversial since then. Some papers have found evidence that yes, there is neurogenesis. Some papers have found evidence that no, there isn't. A lot of it has really turned on exactly how people have looked for neurogenesis. And a key part of doing that has been to look for the expression of certain proteins that are associated with immature developing cells. Now, the problem happens that a certain protein, it might be associated with a developmental cell, but it also might be something that expresses in adult cells as well. So when you use a typical method, which is going to be dissect out a small piece of brain, homogenize it into a kind of brain soup, and then check for the expression of a particular protein, the problem you're always going to have is you have no idea exactly what cells those proteins that you're checking for came from. You have no way of knowing what other proteins were co-expressed with the ones that you're checking for. And really, that's a big limitation because what you're really going to want to look at is not just the expression of any particular protein that's correlated with immature developing cells. In an ideal world, you want to be able to look at the full complex pattern of development, which involves many different proteins interacting with each other as cells go through a series of changes across the developmental trajectory. The problem, of course, is that that's a really difficult thing to look at. Now, luckily, genetic sequencing has come a long way in the last 25 years since that initial hint of neurogenesis in the adult human brain. And it's now possible to actually sequence individual cells and look at their RNA protein expressions. This is exactly what Daniel Franjic, working in the lab of Ninad Sastan, did in a tour de force paper that drew on data from mice, guinea pigs, rabbits, pigs, monkeys, chimpanzees, and humans. Absolutely insane amount of work comparing all of these species. And when they looked at it, they found that they could see the full developmental trajectory of really immature to growing to more growing and finally adult cells. Look at all of those cells along the developmental trajectory in the dentate gyrus granule cells, and they could see the neurogenesis in all of these animals. All of these animals except humans. So we're back to where we were before the 1998 paper. There's no neurogenesis, at least not in the dentate gyrus granule cells of humans. But, as I'll tell you about in this video, this ends up revealing some really interesting implications for what it is to be human and what makes us different from other animals. Hi there, I'm Adam Dede and welcome to a Neuroscience Journal Club, where I bring you a weekly discussion of a recently published article from the world of neuroscience. If you're liking the content, please like, subscribe, comment, all that good stuff. So let's jump right in. Do humans have neurogenesis or not? The approach that Franjic and his colleagues took was sophisticated. They obtained 219,058 individual human brain cells and sequenced every single one of them. What this allowed them to do was look at the full profile of RNA expression on an individual cell level. Then, once they had done that, they took all of these patterns of RNA expression and they reduced it down to a two-dimensional plot, where the axes of this plot have arbitrary units and capture the maximum variability possible in the data. Basically, if you see neurons or cells that kind of cluster in this plot, what that tells you is that they have similar patterns of RNA expression. And so what you can see here is that excitatory neurons, inhibitory neurons, and non-neuronal cells form clear clusters. So that kind of shows us that they're really getting at something important with this clustering method looking at RNA sequencing. And here, these clusters can be thought of as sets of cells that have similar RNA expression profiles. When they plot the same data now, colored by brain region, then we can see that there are certain excitatory neurons that only show up in certain brain regions. For example, this beige cluster is highlighting a set of excitatory neurons that only show up in the entorhinal cortex. Or, plotted another way, the authors applied a hierarchical clustering analysis to RNA expression data to create a tree diagram of different cell types. Now, interestingly, when we look at which brain regions these different cell types were found, we can see that excitatory cells were highly region-specific. Note this section of excitatory cell types that only show up in the entorhinal cortex, for example. By contrast, we can see that inhibitory neurons and non-neuronal cell types were evenly spread across regions. All of this is to say that it's the excitatory cells that really differentiate brain regions. And important to note, those dentate gyrus granule cells, they're excitatory cells, those ones that show neurogenesis in rodents. Okay, so all of this is cool. It shows us that Frangic and colleagues can successfully describe the different cell types that they found when they looked at all of these different human neurons. And that's cool, and probably does actually provide a lot of insight into the variability of different neuronal cell types. But what about neurogenesis? That's the central question we're trying to ask here. So, to try and get at this, Frangic and colleagues did the exact same analysis, but now on tissue that was collected from mice, pigs, and rhesus macaque monkeys. Then they did the same kind of plotting exercise that they did, where they're again plotting on these arbitrary two-dimensional axes, where the axes are capturing the maximum dimensions of variability in the RNA profile, in the RNA expression profile of the different cells. So here you can now see the expression profiles from these different species. You can notice that across species, the sequence cells fall into similar places in the plot. So this indicates that there's a high degree of similarity in the RNA expression patterns across species. However, you can see that that area that's been highlighted with a dotted line in all of the plots, this area of the plot is where the immature granule cells show up. What's cool about this method of analysis is that rather than relying on any single marker of immature cells, we can see the whole trajectory of cell development. You can see they've labeled neural intermediate progenitor cells, NIPCs, and neuroblasts, NVs, which are two stages along the developmental trajectory of new neurons. These two developmental stages appear on the plot as two stages along a pathway that leads directly into the granule cells. You can imagine that this pathway represents the changing pattern of RNA expression that cells go through as they are born and then develop into adult granule cells. And this pathway is clearly visible, leading into the dark blue cluster of granule cells for mice, pigs, and macaque monkeys, but it's notably absent in humans. So next, Frangic and colleagues just repeated the same analysis, but now using only tissue that had been obtained from the dentate gyrus of all the different animals. The idea here was that, well, maybe something's being missed when we take a look at this from the broad view of all of these different brain regions. Let's focus in and look just at the dentate gyrus. When they did this, here you can see evidence of a smooth pathway again leading into the granule cell populations for mice, pigs, and macaques, but again, it's missing in humans. The beauty of this method is that it eliminates the need to specify any single marker of immature granule cells. Again, this is because we're looking simultaneously at all of the steps along the developmental trajectory. Despite this highly detailed picture, there's no evidence of neurogenesis in humans. So one could try and argue that maybe neurogenesis takes a fully different form in humans, and it's just fundamentally different in humans. But why then would it be that all of the other adult cell types look exactly the same in humans as they look in the other animals? It doesn't really hold water. And especially when we think about the fact that they've done such a good job of looking at different animals across phylogeny. We've got mice, we've got pigs, we've got monkeys, all of them showing the exact same pattern, and then humans stand alone. All of this comes together to indicate there's no neurogenesis in humans. So there's no neurogenesis in humans. It's really a pretty wild finding when you think about it. I mean, after all, these weren't small percentages of cells. These were significant minorities of cells that were expressing in the immature state in all of these animals. Specifically in mice, 6.6% of dentate gyrus granule cells were in an immature state. In monkeys, 2%. And in pigs, a whopping 55.6% of their dentate gyrus granule cells were in an immature state. So the immediate question that's going to arise is, why is it that humans stand alone with no neurogenesis in the dentate gyrus? And what does it mean for our cognition and how we think? So to get at this issue a little bit, I'd like to get into the nitty-gritty of exactly how it is that memories are encoded in the hippocampus. And to do this, I'm going to review some findings from a paper published by Thomas Ryan and Dheeraj Roy, working in the lab of Susuma Tonegawa. In this experiment, they had mice, and they fear conditioned the mice to a particular context. To do this, the mice were taken into a small box that they'd never been in before, and then the floor of the box was electrified, giving the mice an electric shock. Obviously, this is a situation that's going to cause the mice to have an intense fear memory for that context. So if they're ever put back in that same box again, they're going to freeze, and they're going to show that they have fear, and in that way show that they have memory for the box. Now, what made this experiment interesting was that the mice had been genetically modified. They'd been genetically modified so that the cells that were active during the fear conditioning experience would be labeled with particular proteins that the researchers would then be able to find later when they removed the brains from the animals. And specifically, they were genetically modified in two different ways, so that there would be two different colors of protein expression that the researchers would be able to find. One of these labeled any cell that was active in the entorhinal cortex projecting to the dentate gyrus in green, and another one labeled any cell in the dentate gyrus that projected onto the CA3 region of the hippocampus in red. And in this way, it was possible for the researchers to identify exactly what cells in these two different connections were active during the fear conditioning. Now, in addition, they also had another manipulation. They injected half of the mice with anisomycin immediately after they experienced the fear conditioning event. Anisomycin is a protein synthesis inhibitor. So what that's going to do is it's going to stop the mice from really forming a new memory. The cells will still become active. The red and green tags will still get activated, and the researchers will be able to find those. But the cells won't be able to form any new synapses or new connections. And in that way, the mice won't really form a new memory. They'll just experience the event, but have no memory for it. And the researchers wanted to see where would activity show up? Where would there be activity that processed the online event of the fear conditioning? And where would changes in the brain be important that would separate the mice that had had protein synthesis and therefore formed a memory from the mice that received anisomycin and did not have protein synthesis and therefore did not form a memory? Okay, so in this image, you're looking at a picture taken through a microscope of the dentate gyrus of one of the mice in these experiments after the brain has been removed from the mouse. The green displays the axons projecting from the entorhinal cortex to the hippocampus that were tagged by the fear conditioning experience. And the two red cells are dentate gyrus granule cells that were tagged by the fear conditioning. And the blue shows us the cell bodies of all of the other granule cells that were not involved in the fear conditioning. The thin red lines that extend up from the two tagged cell bodies are the dendrites of the dentate gyrus granule cells. Where these dendrites pass through the field of green entorhinal cortex axons is where the dentate gyrus cells can take in information being transmitted from the entorhinal cortex. So this showed that the researchers were able to identify what cells were active while the mouse was experiencing the fearful event. Next, the researchers took thin living slices of brain tissue from the mice that had experienced the fearful memory. In these slices, they looked at the dentate gyrus specifically. What they found was that when they stimulated the entorhinal cortex in animals that had not received a protein synthesis inhibitor, the tagged red cells in the dentate gyrus responded vigorously. But the non-tagged, those blue cells in the dentate gyrus, didn't respond very strongly. What this indicated was that the fear memory had increased the strength of connectivity, the strength of response in the dentate gyrus as a result of entorhinal cortex input. So this was an actual change as a result of the fear memory. This is the memory. By contrast, when they looked at slices that were taken from animals that had received protein synthesis inhibition, the anisomycin injection, what they found was that there was no difference between the red labeled cells and the blue cells. That is to say, whether a cell was active in the fearful event made no difference in how strongly it responded to entorhinal cortex input. The anisomycin injection successfully blocked the formation of new memory. Next, they took a look under the microscope at those dendrites, those thin red lines that extended up from the red labeled dentate gyrus granule cells. And what they found was that there were more synaptic connections on the red labeled dentate gyrus granule cells than on the blue labeled cells that were not involved in the fear memory. And this effect was entirely lost in the animals that had received the protein synthesis inhibition. Okay, so taken together, what this tells us then is that increase in the number of synaptic contacts in the entorhinal cortex to dentate gyrus connection leads to an increase in the electrical signal transmission efficacy of the connection. And that is the specific thing that is allowing the mice to form memories of the fearful event. Now, by contrast, when they took a look and they did similar analyses looking at the connection between the dentate gyrus and the CA3, because they had a different colored label that allowed them to separate out these two connections, what they found was that there was no evidence of difference between the anisomycin animals, the protein synthesis animals, and the control animals. So this tells us then that the dentate gyrus synapse onto CA3 is not really an important one for the immediate encoding of a new event, where the entorhinal cortex to dentate gyrus connection, that one is. So we can see this separation where the dentate gyrus is really the key spot that new information is being encoded. And it's not happening so much downstream, even one more synapse downstream at CA3. So to think about how this works, what it means is that at the moment the mouse experiences that fearful memory, the dentate gyrus to CA3 connection has to contain the full range of all possible experiences that the mouse could ever encode, because that connection is not changing as a function of this particular event. You can kind of think about this as every dentate gyrus granule cell to CA3 connection is like a page in the encyclopedia of worldly experience as far as this mouse is concerned. And each one of these connections is a different entry in that encyclopedia. By combining a novel set of encyclopedia entries, the mouse can create a new memory for an event that he's never experienced before. But importantly, in the case of encoding this particular event, he's not making any new entries in the encyclopedia. He's just creating a new lookup index so that he can find a particular set of combined entries. So now, thinking again about the main findings we want to talk about today, this no neurogenesis in the adult human brain, what does it mean when we think about this entorhinal cortex to dentate gyrus to CA3 set of connections and the way memory is encoded? Well, in mice, although they don't use new dentate gyrus granule cells to encode an immediate new memory, because that dentate gyrus to CA3 connection is relatively static, we know that new dentate gyrus granule cells are being integrated into the system all the time. What this means is that over time, the mouse is going to approach the world with an ever-changing encyclopedia of worldly experience. He's got new pages being put in there that allow him to encode new memories in fundamentally different ways. The thought experiment to think about is that a mouse who experiences the exact same event on day 1 and then on day 30 is going to fundamentally have a different way of encoding that memory because of the new cells available in the dentate gyrus to represent it. By contrast, a human who has the exact same set of dentate gyrus granule cells from day to day, year to year, will always encode a new memory in a relatively static way. Now, of course, I'm oversimplifying it. There can be changes in particular synaptic connections that can affect a great deal of flexibility in the system. But certainly, the ultimate flexibility is adding in new cells. So why would it be that a mouse has this dynamic representation system, this ever-changing encyclopedia of worldly experience, and a human doesn't? What's the advantage of that? Why did we evolve that way? Well, one way you could think about it is that it allows us to have an enduring self that persists through time. And this is totally just conjecture, but I think it might make a little bit of sense. That what is a self but a static interpreter of a dynamic world? As soon as you have a situation where the interpreter becomes dynamic, which is what's going to happen when you have new dentate gyrus granule cells being integrated into the memory encoding architecture, then you have a dynamic world and a dynamic interpreter. Well, in that situation, there's no self. There's just a continual whirling system of change, and it's all chaos. Perhaps beautiful, life-inspiring chaos, but chaos. Whereas humans seem to have this unique ability to persist through time. We think of ourselves as being static and constantly interpreting changes. And maybe that has a little bit to do with the fact that we don't have neurogenesis in the dentate gyrus. Anyway, something to think about. That's it for this time. If you're liking the content, please like, subscribe, and comment to try and boost the YouTube algorithm. And if you've been watching my videos for a while, please consider subscribing and supporting me on Patreon, and a link is down below in the comments. Thanks. See you next week.